Hello, everyone. Thank you for joining us today. I'm Jin-Yuan Wang, a senior scientist from STEMCELL Technologies. Today, I'd like to use this opportunity to introduce our microglia differentiation medium system. I joined STEMCELL Technologies about 2 years ago. Now, I'm in charge of a series of 2D neural products, including STEMdiff Forebrain, Midbrain, Astrocyte, and Microglia kits. In this presentation, I will focus on how to generate high-quality hPSC-derived microglia using our products. Hope we can help you with your research. I will begin with a brief note on how PSC-based models can inform and build upon work using animals. I will then touch on the importance of pluripotent stem cell quality for starting downstream differentiation experiments. Next, I will introduce a new product in our line of PSC-based neural models, the STEMdiff Microglia Culture System. I will outline how to use this system and show how to combine them with other 2D neural products, as well as 3D organoids, as co-cultures relevant to disease modeling. Okay, let's start off with a bit discussion around the importance of PSC quality. We know that iPS-derived cells are very useful for disease modeling. This is true whether you are struggling to work with human primary tissue or can't find an animal model that could capture certain disease phenotypes, or you are in need of some human-relevant data to support your path to the clinic. However, I think we can all appreciate that it's not as simple as dropping all the mouse colonies, ordering a vial of frozen iPS, and writing up your manuscript. We know that the bulk of disease modeling, whether it be development, psychiatric, or neurodegeneration, has been done using animal models. This has given us some truly critical insights. For example, in this study, they also used iPS-derived neurons from AD patients and observed a phenotype of decreased mitochondrial area and increased mitochondrial damage, leading them to hypothesize that there could be a defect in mitophage, which they did observe. They went on to investigate the mechanisms of mitophage using C. elegans and mouse models. They found 3 inducers of mitophage reversed memory defects in C. elegans that overexpress human A beta. This is demonstrated by the fact that these AD C. elegans forget that they don't like isoamyl alcohol and tend to move towards it rather than away from it. The mitophage inducing molecules also decrease A beta in this model. Similarly, in mice, they also observed a decrease in mitophage, an increase in mitochondrial damage, and an improvement of memory defects. This is a great example of how you can build a complete story around a human phenotype, complementing human work with animal work to really dig deep into mechanisms. Here is another example. Previous studies using TREM2 mouse model have uncovered its function in microglia survival in AD-associated mutants. However, the role of TREM2 in human microglia still remain elusive. So in this study, they also took advantage of microglia generated from TREM2 knockout iPS cell lines. They examined the caspase-3 and 7 signal in the cells and found that knockout microglia showed increased level of apoptosis compared to control in both normal and cytokine starvation conditions. They also found that phagocytosis of ApoE in wild-type microglia is dependent on ApoE genotype. Interestingly, when TREM2 expression is lost, there would be no significant uptake of ApoE fluorescence above the control. They also used a disease-irrelevant control to test the specificity of this downregulation of phagocytic activity and found that TREM2 knockout would result in substrate-specific defects of phagocytosis. If you'd like to begin your journey to use PSC-derived cells in your lab, you need to make sure you are starting off right. Taking the time to perfect your cell culture technique and ensuring you have high quality PSC is critical to the success of all your downstream applications. The foundation for any successful differentiation is the quality of your starting material. But what are the guidelines for PSC cultural quality? Here I show two examples of publications highlighting some of the issues. One by the New York Stem Cell Foundation and one by a large group of stem cell banks and international consortia led by Global Alliance for iPS Cell Therapies. They touched on many themes, but one that applies to all hPSC research is this idea of importance of quality control, verifying iPSC properties, and extensive characterization to ensure valid and reproducible data. Because phenotypes across diseases can be variable, reproducible data are essential to detect meaningful differences between groups. This is why it is really important that cell quality attributes need to be considered by everyone working with PSC, not just those researchers moving towards the clinic. However, we can't just spend all of our time and money testing cell quality. So there needs to be a balance with what we can do easily and routinely. At stem cell technology, in line with this quality discussion from key consortia, we have identified four aspects of pluripotent stem cell health that can be routinely monitored: morphology, gene and marker expression, functional pluripotency, and genomic integrity. This is why whenever we are developing a new reagent or protocol, we design our culture system to reduce cell stress, increase reproducibility, and cell quality. Additionally, we have designed tools to assist our customers to assess these critical quality attributes on their own cell lines. Some examples are the hPSC genetic analysis kit, which measures common amplifications and deletions found in PSC culture by qPCR. The tri-lineage differentiation kit, which allows you to differentiate your cells to derivatives from all three germ layers, and our antibodies and qPCR panels that allow you to characterize your cells in terms of expected marker expression. STEMCELL provides our family of TeSR medium to ensure you can maintain high quality human PSC. This includes our newly released mTeSR Plus that allows for reduced feeding without reduction of critical quality attributes. This was performed through stabilization of key components, including FGF2, as well as enhanced buffering capacity to combat medium acidosis and reduced cell death. Shown here are examples of the excellent colony morphology you can get in TeSR media compared to using a suboptimal media. Note the smooth edges and consistent cell morphology within each colony. On the right, you will see an example of the very high percentage of OCT4 and TRA160 positive cells as determined by flow cytometry. Further, we offer the convenient tri-lineage differentiation kit, which you can use to assess functional pluripotency. Cells cultured in TeSR media have a high efficiency of differentiation to all three germ layers, as measured by flow cytometry of typical markers for each layer. The bottom table summarizes the results of genomic integrity tests over several passages. No amplification or deletion are detected using our PCR-based genetic analysis kit every 5 passages, correlating with a normal G-binding karyotype at P20 to P30. Once you are confident you have good quality starting material, then it's time to start thinking about the downstream cell types that you want to generate. At STEMCELL Technologies, we have easy to use, reproducible kits to generate forebrain neurons, dopaminergic neurons, astrocytes, neural crest cells, and organoids. These kits are all based on established methods for echodermal differentiation. However, there's one cell type of brain you can't get using this technique. Microglia, the brain resident immune cells, are mesodermal in origin, and the protocols for differentiating these cells have only been published recently. Microglia are critical for normal neuron health and development. They are also responsible for brain's immune response. As such, you must consider microglia function for any study looking at brain development, infection, or inflammation. The key step to producing PSC-derived microglia is first generating the right kind of hematopoietic precursor cells. Microglia originate from primitive hPSC in the yolk sac, and that is exactly the kind of primitive progenitor that is produced using our STEMdiff Hematopoietic kit. After harvesting the cells on day 12 of the protocol, you can seed the cells into our upcoming microglia culture system. As you can see in this table, STEMdiff Hematopoietic kit can generate hPSC with high purity across multiple cell lines. After the harvest of hPSC, we switch the medium to STEMdiff Microglia system. This system contains a 24-day differentiation phase, followed by 4 to 10 days of maturation. Handily, we have found that you can freeze down hPSC from day 12 of the hematopoietic kit and proceed with microglia differentiation later. Or you can freeze down microglia at day 24 and thaw them later into maturation medium to complete your endpoint experiment. At the end of maturation, the majority of cells you produce will express the expected mononuclear cell markers, such as CD45, CD11b, and TREM2. On the left, you can see some representative flow plots for all these markers from cells cultured in mTESR1 or TESR-E8 media. The right table shows the average percentage of these markers over at least 3 technique replicates across several ES and APS lines in various starting culture media. We also examined the expression of microglia markers such as EBO1 and PO1 by ICC. Magnified images show typical small microglia morphology. To set your mind at ease that you are actually making microglia and not other mononuclear cell type, we directly compare our microglia to macrophage and monocyte. In this plot, we are showing the CD14 mean fluorescence intensity is much higher in PSC and blood-derived macrophage and monocyte compared to the CD14 intensity of microglia derived from 4 different PSC cell lines. We went on to look at CD206 and CD209 expression by flow cytometry and found that we could detect co-expression of these typical M2 macrophage markers in about 40% of our M2 activated blood-derived macrophage, but, but not in M1 activated macrophage or in PSC-derived microglia. Then we checked a series of typical genes expressed in microglia. As we can see in this figure, microglia produced with our kit show significantly different expression profile compared to monocytes. While the expression patterns of these genes are more variable, we see quite a few genes with different expression level compared to both M1 and M2 activated macrophages. When we compare RNA sequencing data from the microglia produced using our system to the published RNA sequencing data from the publication on which the kit is based, we see that both PSC-derived microglia types clustered together along PC1 and separate from monocytes and fetal microglia. In fact, when we gathered all of the published RNA sequencing data across multiple protocols and publications, batch corrected it, and compared it to our in-house RNA sequencing data, we found that all the PSC-derived microglia grouped together across PC1 and PC2, and away from most of other mononuclear cell types. From this, we hypothesized that the STEMdiff Microglia Culture System can generate the type of cells you are looking for, regardless of your starting protocol of preference. Importantly, these microglia-like cells are functional and capable of phagocytosis, as shown on the plot, using macrophage as a positive control, and the parent PSC lines as a negative control. All of our PSC-derived microglia lines are capable of phagocytosing bioparticles. On the right, you can see a representative image of microglia labeled in green, gobbling up disease-relevant peptides like human beta amyloid fragments. We also treated these microglia with LPS and found the secretion of expected cytokines as shown in this figure. Now we are able to generate neurons and glia from PSC with our products. Next, I will introduce how to combine these cells together for disease modeling. As we know, not only neurons, but also other types of cells in the nervous system play a critical role in the pathology of neurodegenerative diseases. So single culture of certain types of neural cells may not be able to capture the disease pathology accurately. In order to have a more physiological model and to study the interaction between neurons and glia in the context of diseases, researchers have developed co-culture systems to mix different types of cells together. The idea is to generate neurons and glia from either health control or patient-derived cells, then mix them together to investigate the role of each type of cells, as well as the interaction among them. These are the Forebrain-Type neurons generated by our kids. After 2 weeks in maturation media, you can expect over 90% TUG1 positive neurons and less than 10% GFAP positive astrocytes. And the majority of them are Forebrain-Type, indicated by FOXG1 staining. These are the astrocytes generated by our kids. As we can see from these images, our products can help you get over 70% S100 beta positive and more than 60% G5 positive astrocytes with less than 15% double coating positive neurons. This is the workflow of neural astrocyte co-culture using our products. It's very straightforward and easy to use. Our data show that forebrain neurons and astrocytes produced by our kits can be cultured together with our forebrain maturation medium. Interestingly, neurons with astrocytes had longer neuroid outgrowths, as well as better survival rate compared to the control group. This is the workflow of Neuron-Microglia co-culture using our products. It's also very straightforward and easy to set up. Our tests showed that the forebrain neurons and microglia can be cultured together with modified brain phase neural medium. Here we use EMBO1 to stain microglia and TUG1 for the neuron. We also tried to mix forebrain neurons, astrocyte, and microglia together. This is the workflow of triculture. Basically, we did the neuron astrocyte co-culture first, and then seed microglia onto them. After the triculture, as we can see, these 3 types of cells can live together, as indicated by TUG1 for the neuron, GFAP for the astrocyte, and EMBO1 for the microglia. Interestingly, if we introduce mechanical injury into the in vitro culture, then the triculture group showed increased astrocyte motility. We found more G5 positive cells in the scratched area of the triculture group. Now, I have shown you how to generate 2D co-culture with neurons and glia generated by our kits. I will next show you how to do a 3D co-culture with our products. Here is an example of some of interesting ways that you can use 3D co-culture for modeling inflammation. In these two papers I highlight here, the authors used PSC-derived microglia to demonstrate how injury to brain organoid results in microglia taking on a pro-inflammatory phenotype. The authors of the second paper, shown on the bottom, showed a similar response to Zika virus infection. In the next few slides, I will show you how you can replicate the results of the top paper. First, let me introduce how we generated the organoids that we used for this experiment. Very shortly, we have a kit available for pattern organoid generation based on Sergio Pasca's protocol, including the dorsal and the ventral forebrain. This protocol takes advantage of the agri-wall system, making it amenable to scale up and is matrix-free. Once the EEBs are formed in the agri-wall, they are taken out and put in static suspension culture for the duration of expansion, differentiation, and maturation. These media are optimized for increased reproducibility and robustness across multiple cell lines. We generated dorsal forebrain organoids, which contains multiple putative cortical areas with a radial arrangement of PAC6 positive progenitor cells surrounding a ventricle-like region. MAP2 positive neurons migrate out from these regions and display layering of deep and upper layer neurons, mimicking cortical development in vivo. As in normal brain development, gliogenesis happens after neurogenesis, and so that in dorsal organoid, we see astrocytes forming at later time points. As early as day 50, we believe the presence of astrocytes in brain organoids of any kind is critical for microglia survival and incorporation, which makes sense as traditional primary microglia are best cultured in astrocyte conditioned medium. This is how we set up the co-culture. Basically, we generated brain organoids and microglia from PSC using our products separately. Then we put them together with our medium and continued the co-culture for a week before the downstream tests that you want to try on the culture. Here we use the needle to generate injury site on the co-cultured organoids. This figure shows that our microglia survive and integrate fairly well into the organoids. The majority of cells take on a resting morphology as indicated by the processes shown by the white arrow. On the right is our injured organoid. As you can see, in response to injury, the microglia are taking on a more amoeboid morphology, characteristic of an inflammatory response. We're still working on characterizing this inflammatory response in pure microglia cultures and co-cultures. And if this interests you and you'd like to explore this with us, we're happy to discuss collaboration. Here are some potential projects that we'd like to work on in the future. How do PSA microglia affect neuron firing in culture? How do diseased microglia affect neuron health in co-culture? Can we replicate disease phenotypes and dissect out cell autonomous or non-cell autonomous mechanisms? Do PSC microglia follow an M1-M2 activation phenotype? We are open to discussion and collaboration if you are interested. To sum up today's talk, I'd like to reiterate how critical knowing the quality of your PSC is before starting any kind of expensive and time-consuming disease modeling project. Carefully consider what model is most appropriate for your research question, and please don't hesitate to reach out to us for recommendations. If you are looking to do more complex 3D and co-culture models, we'd love to hear from you to discuss our latest results and dream up what we could do next in order to make this system accessible for everyone to use. Here are some resources that are related to today's topic. You can find more on our website. Hope this can help. That's the end of today's presentation. If you have any questions, feel free to contact us. Thank you.